Good morning, everyone. Thank you for coming to today's seminar. Also, thank you for the people online. For you online, please leave us a message if we hear as well, or if you have any other technical issue, and leave your questions on the chat. Today, we have the pleasure to have Frederic Vagneron present his work, and I will ask Rémi Fouissard to introduce him.
Thank you very much. So it's a pleasure to have Frederic coming here to present his research. So to present him, he has a very nice website at the University of Strasbourg. So he did his PhD on the influenza. And it's called Aux frontières de la maladie, l'histoire de la grippe pandémique en France entre 1889 et 1919. He got several awards with this writing. Is it true? So I presume it was published, or it was published as a PhD, or it was also published in
Articles.
Okay, articles. And then he did a postdoc in Zurich, also on science history and medicine history. And now he's working in the University of Strasbourg as a historian. And he's working now on at least 3 projects that are concerning the innovation and also the antibiotics between the years of the 70s to 2010. This is one project. Another project is the history of health in Europe between 1945 and 1960. And the third one is a new project is how to live with mosquitoes in the region of Lorraine. So thank you very much for coming, and the floor is yours.
Good morning, everyone, and thank you for the introduction and for inviting me today. It's a pleasure to be with you today, even if it's a bit of an adventure for me. I'm an historian of medicine and public health, and this project on culture collections pushed me into the new territory of the history of biology. I'm not a biologist by training, but general historian. Coming from the history of epidemic, especially influenza, I found myself rethinking many assumptions shaped by years spent on the medical side of early bacteriology at the end of the 19th century, which at the time was also tackling issue with viruses because I didn't know actually what were viruses. Today, I want to introduce you to 3 connected stories. First, the biography of a somewhat forgotten bacteriologist and curator, Paul Hauduroy, was born in France in 1897 and died in Switzerland in 1967. Second, the history of an institution in Lausanne, the International Type Culture Collection, which briefly became the coordinating center of an international network of microbial collections. And third, the story that is related to that of an episodic event, the myxomatosis, the European myxomatosis episodic, which unfolded through this very infrastructure of circulating microorganisms. So to use a microbiological metaphor, what I'm trying to do is to culture.
The presentation is not shared, so people will see.
Oh, okay. But at least they haven't missed anything.
No,
Not yet. So what I'm trying to do is somehow to culture this biographical and institutional sample, Hauduroy and Lausanne, in different enriched media, different historical context here, since I'm an historian, so that its full significance becomes visible. My goal is not to isolate scientists, political or the biological, but to examine how they interact. And Hauduroy is a perfect case for this. What Hauduroy and the Lausanne collection reveal is this. They, and he was not alone, contributed to the early construction of an international microbial infrastructure. they inadvertently triggered one of the first open-air release of microorganisms into the environment, a story I will return to in a moment. And they challenge in their own way the unstable boundaries of the microbial world at a time when that world was expanding dramatically with phages, viruses, plasmids, and eventually recombinant genes, complicating the story of what a species was. So today, I want to take you into the history of something that quietly supports much of modern ecology, microbiology, and biotechnology, microbial culture collections. These collections may seem like invisible technical resources, places you order strains from, but historically they were and still are also scientific centers, geopolitical tools, economic assets, and places where nations negotiate access to microscopic life. To make this story concrete, I will follow the early history of this specific collection, the Lausanne collection. Lausanne played a surprisingly central and short role as a hub for international cooperation, standardization. It was a UN-funded cooperation, and later a symbol of the struggles and limits of the first attempt to build international collaboration, which ultimately shaped what was created in 1970, the World Federation of Culture Collections. So the main questions I want to tackle with you today is how is the knowledge infrastructure on microbial culture collection built on an international scale in the mid-20th century? And what is its connection to the WFCC? What kind of history of microbiology does Hauduroy's trajectory allows us to tell? And by that, I mean beyond the usual narratives of the great heroes of the bacteriological revolution at the end of the 19th century, or the molecularization associated with the boom in molecular biology and genetics in the 1960s. The relationships, agreements, procedure, and containers that were produced at the foundations of the infrastructure are somehow still alive today, although contested. It is a long-lasting heritage which still constrains the practice of microbiology. So just briefly, a word on this concept. We are historians and social scientists of infrastructure of knowledge. So here I follow a line of questioning now well established in social sciences and social science and science and technology studies. And one key reference, for instance, is Starr and Walker, who've been working on the history of the International Classification of Diseases, the ICD. So simply put, infrastructure studies imply a social technical approach looking not only at the material side of the work, but also at the institutions, cultural and legal norms, social relationships, and everyday routines that provide the foundation on which standardized procedures, in this case for running and maintaining culture collections, can emerge and are maintained despite often being invisible. So my goal is telling the story of a promising project, a new international infrastructure, with a central bureau of standards, so to speak, and how it ended up in a complete state of oblivion 3 decades later. This location, as I said, was located in Lausanne, Switzerland, and created in 1944 within the Bacteriology and Public Hygiene Institute, led by Hauduroy, since 1940. The fate of the Lausanne Collection Center is all the more intriguing that it happened in a very promising period of time, decade after the Second World War, that were shaped by internationalism in the medical and biomedical fields, with new projects and infrastructures supported by flagship new institutions such as the WHO, or maybe more surprisingly, the UNESCO. So here is the brief outline of the talk, and the material I'm going to use are archival material from Hauduroy that are right now in Lausanne, but also documentation and archival material from the American Society for Microbiology that are stored in Baltimore, UNESCO archives in Paris, and gray literature, so to speak, and scientific publications. So my first part, this part, despite their growing importance for the bacteriological discipline and its pure cultural mantra, early culture collections rested at the beginning of bacteriology, medical bacteriology mainly, on fragile foundations. Between the 1890s and the beginning of the 20th century, important collection of microbes were founded in Prague, la collection Prague, in Paris at the Pasteur Institute, and in other places in Europe mainly, for both research, teaching, and sometimes exhibits. These collections were fragile microbial museums. The word museums was actually used at the beginning. They preserve strains by constant subculturing. Strains could mutate or disappear, and collections often collapsed when curators retired. And that explains why the collection, or part of it, was lost, and another part of it went to the US. Bacteriologists could deposit curious or valuable cultures to these collections and request strains with curators beginning to produce catalogs of holdings from around the beginning of the 20th century onwards. Funding of these collections was erratic, and collections maintenance often depended on individual workers, often women, with little continuity. Rudimentary knowledge of type variation and artisanal conservation practices implied that collections' contents were also far from stable, especially before the use of dry freezing technologies in the 1930s. After World War I, the growing involvement of states and major non-governmental funders in securing microbial resources emerged, especially the non-profit American Type Culture Collection, ATCC, which was founded in Washington in 1925, and in the UK, the National Collection of Type Cultures at the Lister Institute in London, at the same time, just right after the First World War. At the international level, Ralph St. John Brooks was the curator of the NTCC in England, and US bacteriologist Buchanan assumed leading roles on the newly formed taxonomic subcommittee of the International Union of Microbiological Societies, created in 1930 on the occasion of the first International Congress of Microbiology that was held in Paris. In a sense, it was a shift in microbiology from a Franco-German to an Anglo-American leadership. Anglo-American bacteriologists used taxonomic standards such as the one of the American Society for Microbiology and the Berger's Manual, which first edition was published in 1923 and called Berger's Manual of Determinative Bacteriology, not systematic bacteriology yet. And service culture collections came to be thought as invaluable repositories, recognizing them as much of the future development of bacteriology that will depend upon their adequate growth, support, and utilization. And that's a quote from the International Congress of Bacteriology. The link between pure culture and classification was reinforced in the post-war period when the deposition of pure type cultures in collections, similar to type specimen in botany or zoology, was stipulated as a requirement to validly publish new bacterial species. So here comes Hauduroy in the story. Born in 1897 in France, in Montpellier, Hauduroy was a promising medical bacteriologist. He earned his PhD in 1921 in Strasbourg under the Pasteur Amédée Borel, and worked closely with Félix Derel, and became an early investigator of bacteriophages, studying them in hospitals and in the environment too, to understand their nature and therapeutic potential. During the 30s, even without a proper academic position in Paris, he published widely, including on the nature of viruses, a really new topic at the time, and co-authored a well-known Dictionnaire des bactéries pathogènes in 1937. And in 1936, at the occasion of the Second International Congress of Microbiology in London, he presented, for instance, a paper on filterable bacteria, a subject that I will come back to at the end of this talk. It was around this time that he first contacted Ludwig Rachmann, head of the League of Nations Health Organization, prior to the WHO, Polish scientist, raising concerns about the chaos of bacterial nomenclature and calling for a standardized international body, like the one that was working on the Salmonella at the time, to elevate the scientific status of microbiology worldwide. In 1940, as I said, he was appointed professor of microbiology at the Lausanne University and became head of the Bacteriology and Public Health Institute. And with the war, Hauduroy's project in Lausanne gained legitimacy because it could act as a kind of neutral broker, keeping scientific links alive between laboratories suddenly separated by the war. Being in Switzerland, it was shielded from the direct pressures, damage, and financial strains that affected so many other institutions. So, in 1944, and in the years following the Second World War, the Lausanne project had a very good start. It was a local initiative from the Swiss Academy of Medical Science, and the goal was clear: collecting and cataloging all microbes and viruses possible with a special focus on pathogens. The funding was rather limited, 10,000 Swiss francs by this Swiss Academy. And this project has nevertheless the support of a great deal of experts in its international scientific committee under the direction of Ralph St. John Brooks from the English Culture Collection, and with a handful of celebrities such as the Danish bacteriologist Madsen or Sir Alexander Fleming. The new center soon received the official support of the newly created UNESCO, which developed, maybe a bit surprisingly, even the S of UNESCO is for scientific and science, sorry. And UNESCO developed its own agenda in the field of bacteriology. The UNESCO head of science division, Joseph Nidam, followed the proposition of a Brazilian bacteriologist, Da Fonseca, and that's the presentation in the middle, the slide in the middle, to create an international collection center with the aim of pursuing peace and development through scientific research. And in 1948, the Lausanne Center became officially the chosen center. In 1947, the professional bodies to embrace the cause of this center in Lausanne during the 4th International Congress of Microbiology in Copenhagen, the first after the Second World War. And a new International Federation of Culture Collection of Microorganisms was created on this occasion within the International Association of Microbiologists, the IMS, if I remember well at that time. The creation from scratch of this International Center for Type Microbial Collection in Lausanne was so supported by the UNESCO, which provided funding channeled by the International Federation of Culture Collection Executive Committee to national collection of particular value or endangered. They still have in mind the story of the crowd collection, for instance. This advisory committee was staffed with first-class bacteriologists and taxonomists of the time, and you have a list of them here on the slide. The idea was that Lausanne would coordinate a worldwide network of representative collections, making microbial resources internationally accessible. This was the high point of an internationalist optimism. And Lausanne became a hub for a vision of equality in science, a place for shared taxonomies, standardized collecting, and through Hauduroy and St. John Brooks and the IFCC, it was working on the very first catalog of culture collection in the world. So, among the many activities the center carried on as part of the infrastructure, it was, of course, research activities and technical meetings focusing on the actual work in the laboratory on standardization of culture, for instance. Publication of various scientific books and manuals were also happening at the time to foster this collaboration between scientists of the world. And Hauduroy was also traveling in different countries to present Lausanne's work in Europe mainly, but also in the Soviet Union, for instance, in 1956, and Japan in 1951, to explain what was the work that was achieved in this Lausanne center. Led by Hauduroy, the center started its work with a handful of assistants and employees. And Hauduroy published a couple of manuals to explain, mainly in French, the standard procedures and techniques to identify, cultivate, and ship specimens and propose type microbes to Lausanne. This work was following the rules of the pure culture era, and I borrowed this expression from a colleague, Matthias Broad, that was born not without discussions at the end of the 19th century. To quote Hauduroy's book, the goal was clear. If we want it, in precision must no longer exist in microbiology, and standardization of practices was apparent at both the technical level and in paper. At the technical level, Hauduroy was a promoter of the dry freezing technique, lyophilisation, that was invented at the end of the 19th century for food preservation. His work as a collector of strains and type microbes was coupled with an experimentation work to improve the stability of the dry freezing method to avoid any contamination in the storage and allow the organisms to be preserved and undamaged for researchers. At the paper technology level, Hauduroy and his colleagues were spending a lot of time and also a lot complaining about it to trace each strain that was sent to its first publication in order to create a standard catalog. That was the first goal of the institution. The work of nomenclature was not so popular at the time in the field of bacteriology. Many doctors were sending their strains, so it was dominated by medical bacteriologists. So despite a chronic lack of personnel, the Lausanne Center achieved a rather impressive work, considering that there was nothing before 1944. This work was also to act as a platform of exchanges of both information and specimen. In 1952, on the left, for instance, the annual report mentioned the shipping of 900 strains all over the world. 200 were received and stored. And at that time, Hauduroy starts an international inquiry to locate and collect viruses around the world. A decade later, the Lausanne Center is free shipping more than 1,200 microorganisms per year, answering requests and still dry freezing strains. But most of these strains are circulating in Europe, in fact, with only, for instance, 50 strains from the US in 1963-64. At that time, the activities of the center have reached their peak. The years to follow, the activities are slowly declining, and it will be such until the end of the decade. In the 70s, as I will explain, the center actually lose its international function. So Lausanne became a place well known in the bacteriological field when it comes to preserve strains of particular importance. And just an example, here you have an example of the Canadian medical expedition to Easter Island that's been studied by a colleague, Jacqueline Delphine, that's the cover of her book, and showing that the main medical bacteriologist, Georges Nogrady, actually sent some salt sample and blood serum to Hauduroy, again, in this idea of sharing and maintaining these strains of specific value. The idea actually was that there was a project to create an airport on Easter Islands, and so they wanted to preserve what was there before there were any damage. The airport was never actually established. One crucial part of the Lausanne Center was the collection of people, so to speak, not only strains, culture, or information. And this activity seems to be more than difficult to maintain in the long term. It's difficult to single out one reason. Was it a question of person and relationship, of a common language? And through the archives, I have the impression that Hauduroy was not really fluent in English. The fact that Lausanne was a rather small research place, still within the Bacteriological and Public Health Institute. Or maybe the competition with other projects. At the time, of course, molecular biology was on the rise. A good proxy to figure out is the circulation and the discontinuity of the newsletter of the center. Its first issue starts in 1949, and many issues are following until 1952, 53. Then it becomes a very sporadic publication, even if this discontinuity comes to an end when the WHO in 1961 provides a special grant. But at that moment, beginning of the 60s, it seems that the connection between the center, its director and curator, and the scientific community of microbiologists was loosened. In fact, despite the newly created UN agency UNESCO endorsement, the International Microbial Tab Collection in Lausanne and the International Federation of Culture Collection did not last long as the main center of the new international infrastructure of culture collections. Early on, in the 1950s, some clouds appeared, so to speak, and multiple rivalries and obstacles occurred. Among them, for instance, and from the very beginning, the competition between the WHO and the UNESCO. And here you have a letter from Nidam, who was the head of the science division at the UNESCO, who was acknowledging this competition between the WHO, who was not very, and the UNESCO, and the WHO was not very keen to see a new international body working, especially on type culture collection, because they thought that they had like sovereignty over issue of medical importance. Another issue was the competition with seemingly collaborating centers within the network, and especially the American type culture collection. And here you have an example of American microbiologist Steinhaus, who went to Lausanne during one of his trips and was not exactly warmly received by the assistant of Hauduroy because of its presumed connection with the ATCC and the policy that was used by the ATCC in making scientists pay for the strains that they were asking from the ATCC, which was not the policy that was supposed to work within the infrastructure. Another example that will be of much more importance was the dual role, so to speak, of the Commonwealth and this kind of silent clash between the British and Commonwealth culture collection and the Lausanne Center. As Cowan put it, and he was the curator succeeding St. John Brooks at the head of the NTCC, the refusal of the different Commonwealth culture collection was a blow and an obstacle to the completion of the main task of the Lausanne Center, which was this catalog of all the culture collection in the world. At the same time, the International Federation of Culture Collection entered into a crisis in the mid-50s due to UNESCO's reduced funding for collection and internal conflicts between the AFCC and the EIMS, the International Association of Microbial Society. So by the early 60s, many microbiologists recognized the lack of coordination between the collection as a major problem, and it was the beginning of the demise of the Lausanne Center. And in particular, Canadian, Australian, and Japanese microbiologists and their national culture collections used the UNESCO to reassemble the infrastructure of culture collection. And in so doing, it was the relegation of the Lausanne Center and Hauduroy. Instead of using a central information center such as the one in Lausanne, the idea was to create an international information network linking national collections. This also mirrored a shift toward developmentalist policies at the UNESCO in the field of applied macrobiology. Once regarded as a key reference center within the international infrastructure, the Lausanne Center became highly coveted for its data, especially what had been produced 40,000 microorganism identification cards that had been collecting since the beginning of the collection in Lausanne, and that were now on punch cards through an IBM system. So Hauduroy tried to resist its demise and carried on the international center functions in the 60s, still working on technical assistance and international collaboration. He's tried to restart the World Catalog Project in collaboration with the new head of the ATCC in the US. He tried to revive his connection with UNESCO, and also started to have a new contract with the OECD and look for support from the WHO that helped, for instance, the publication of this informational newsletter. But within less than a decade, a new federation was set up through various scientific meetings, first in Toronto in 1962, in Moscow in 1966, and the Mexico International Congress of Microbiology in 1970. In a sense, Lausanne Central Information Service foreshadowed modern digital databases with its IBM data system. But that's the World Federation of Culture Collection and its World Data Center that will be created and installed in Brisbane in Australia in 1970, that inherited this logic with one famous bacteriologist, Victor Skerman, as an important new actor in the promotion of a new numerical taxonomy supported by SNIS in order to integrate more features in the process of checking relatedness between the microorganisms. So ultimately, the center of Lausanne died almost with Hauduroy, who died in 1967. All things considered, Lausanne appears at key turning points at the site of the first international federation and UN-funded Central Information Service, as a symbolic test case for the limits of internationalism. And I showed the competition between different national culture collection. And as an example of how international geopolitics shaped scientific infrastructure at the same time. There were also international factors leading to the ultimate failure of the Lausanne Center project, because this center was actually not a national culture collection for Switzerland before the 70s. And the support of the Swiss Academy was really on this international project. There were also competition in Switzerland with other funded biomedical projects and the lack of funding for this specific collection center. And the new director of the Institute of Bacteriology, Valentin Boniface, was trained as a molecular biologist and was not really interested in following up the work of Hauduroy. So in short, it was an institution heavily dependent on its founding father. Maybe one of the major achievements from the first work of Hauduroy and then the new World Federation of Culture Collection was the publication in 1980 of the approved list of bacterial names, co-edited by Skerman. This work actually consolidated decades of conflicting taxonomy, reduced tens of thousands of names to about 2,300, and established a clean start date for new names in January 1st, 1980. And this approved list of bacterial names relied heavily on identifying preserved type strain in collection with names that would now be validated by the Judicial Committee of the International Committee for Bacteriological Nomenclature. So somehow Lausanne's earlier dream of a global catalog and a new reduction of the chaos in the naming of bacterial strains was finally achieved. So in the second part of the talk, I would like to develop another idea and another take of this circulation and infrastructure allowing the circulation of microorganisms. The new circulation allowed and developed through centers such as Lausanne also worked the other way, so to speak. In other words, the infrastructure itself had wider consequences beyond its international operation, even for a larger environment. One incident, to not say a catastrophe, made the Lausanne Center famous internationally, despite the attempts of Hauduroy to remove the public and, I guess, scientific discomfort. In 1952, Hauduroy sent to an old French bacteriologist Myxomatosis strain that had just been collected in Brazil by a veterinarian from Lausanne and stored in the center. A year later, the Myxomatosis epizootic, which will kill millions of rabbits in Europe, was traced to Lausanne, and the release of this highly virulent strain by Armand de Lille, so this Pasteur Institute researcher. The strain was then named Lausanne strain after its origin. This wild epizootic contrasted to the controlled infection of rabbits led by a famous virologist in its team, Frank Fenner, in Australia, right at the same time to eradicate the rabbit pest in Australia. And on the left was actually a receipt of the copy of the mail that Hauduroy used to send the strain to Armand de Lille in France. This release became a top news in all Europe because of the consequences, of course, for the rabbit population, but also for many people working in the fur industry, hunters, and first activists for the conservation of nature. A major crisis that led to several trials in France that Hauduroy tried to escape. This incident was a major blow to the center prestige, one can guess, but it also became a major ecological experiment that led to hundreds of publications about the dynamics of epidemics between hosts, parasites, and environment. That episode also participated to framing of new rules in the shipping of biological material through the Postal Union Conventions of 1964, a decade later. So, of course, Hauduroy kept trying to distance himself from this event, as his personal archives clearly show. And here, in a news magazine, Illustré, 10 years later, we see how the center tries to present itself as not only modern, but also safe, essentially erasing the memory of the Myxomatosis event. During the two trials that began in 1954, involving Armand de Lille and the plaintiff whose rabbits had been destroyed, Hauduroy's responsibility was dismissed because of the instruction he had given to Armand de Lille in terms of the proper use of the strain. In the end, Armand de Lille was found guilty, yet he later received the Agricultural Merit Medal, la Médaille de Mérite Agricole, from the French Ministry of Agriculture. I won't go in details here, but this case became a much studied experiment of host parasite co-evolution, including the role of vectors under different climates. And the Australian campaigns of eradication led by Frank Fenner, as I told you, asked in 1954, the letter, two letters that you have from the archive in Australia, So they asked the Lausanne Center for the Brazilian strain to use it in the field against their own European rabbits that were in Australia, comparison to the South American rabbits who were from another species. As Fenner puts it at the bottom of the second letter, what was regarded as a curse in Europe is a boon in Australia. The most preeminent scientific legacy of the Lausanne Collection Center was in many ways this wild and live experiments, so fruitful for researchers around the world, especially working on population ecology. The virus had been recently isolated in Switzerland from an infected Brazilian animal, and within months it spread by mosquitoes vector and also the European rabbit fleas. And the virus began to move rapidly across France, killing between 35 and 45% of those animals infected. However, by 1955, scientists began to detect attenuated strains of the virus, and despite occasional outbreaks of high virulence and very large overall mortality due to the disease, there was thereafter a decline in the mortality. Though long after the initial outbreak in France, the virus spread, for instance, to England, maybe intentionally with the help of farmers or foresters. And finally, the Lausanne strain made its way to the culture collection infrastructure, the American type culture collection, and was used decades later in phylogenetic studies. Especially to work on the evolution of virulence after its release in the 1950s. So that was another legacy of this infrastructure of culture collection. In this last part, quickly, I want to look at the infrastructure of culture collection and Hauduroy in a third and different way. Because the Lausanne Center was intended as an international repository of type specimens, it fits neatly into the usual narrative about the shift from early bacteriology, driven largely by medical practitioners, to the later rise of general microbiology, which was more inclined to adopt an evolutionary approach to classifying microorganisms. As such, the medical bacteriologist Hauduroy and his colleagues involved in the emergence of this international infrastructure would share the gold standard of the early days of bacteriology, such as what they call the monomorphism idea, rejecting bacterial variability, the necessity of pure culture to identify type specimen through morphology, and later, of course, other features such as growth, staining, nutrition, etc. But a new historiography has recently shown to what extent early microbiological practices were more diverse than what had been taught. Enriched, so to speak, by the new perspective of microbial ecology. This has led also to a re-evaluation of the role, function, and practices of culture collection for scientists at the time. In fact, scholars such as Victoria Lee on the story of Japan microbes and fermentation, Charles Colmer on various members of the Delft School in the Netherlands, or Matthias Groth on Winogradsky and other soil microbiologists, have shown a wider range of domains where original microbiological practices happened early on, often contesting the dogma of pure culture and the dominance of medical practitioners over bacteriology. In a similar way, as these other scientists, Hauduroy was both a curator of type specimen culture, as I presented, writing orthodox, so to speak, handbooks or manual technique microbiologic, and an original researcher working at the boundaries of microbial species definition. Here are some features of his career and some publication. As we see, a large scope of different topics going from what they call the ultra-virus, new topic at the time, especially in the interwar period, but also bacteriophage, and this question that I'm coming to now, filterable bacteria. As Amsterdam's colleague has shown, arguments about whether or not bacteria undergo cyclic changes of form and functions played a significant role in the pre-World War II investigations of bacterial variation, largely in contradiction to the monomorphism theory, this idea of the stability of species developed by early microbiologists such as Kohn and Robert Koch. Articles on bacterial variation were more than common in the 1920s and 30s. And the early study of viruses and bacteriophages in the 1920s was also fostering this line of research because of the enigma of the still invisible viruses or the lytic action of bacteriophage on bacteria. And after World War II, the action of new antibiotic substance on cell wall bacteria also triggered new research. And in all these areas, which was some kind of unorthodox at the time, Hauduroy was involved. Hauduroy was one of the French representatives of this line of research, and later also, of course, in Switzerland through the collection. And his initial work on filterable forms of bacteria started in the 20s, but he gained a new dimension with its new participation in culture collection management in Lausanne. I'll be quick because I'm running out of time now. Hauduroy started, for instance, a close correspondence and research with Amy Klinenberg-Nobel at the Lister Institute in the 1930s. I've made a bad picture on the right of the slide, who discovered in 1935 what she called the L-form bacteria, Lister-form bacteria, and later L-phase bacteria, which were and are presenting more or less stable lack of cell wall. The work they carried out over 20 years of collaboration, exchanging strains, sharing laboratory practices, and meeting at working sessions such as the one in 1956 in Lausanne that was related in nature, thus led them to investigate difference between this curious form of bacteria and, for instance, what would be called in the 1960s mycoplasma, microorganisms without cell wall that were called at the time propulmonal-like organisms, PPLO. So as such, what is interesting to me here, looking in another way to this culture collection network, is that you can actually have another look of what was the mainstream work on pure culture at the time. Maybe to wrap up, I just want to highlight that these debates, for instance, the case of Hauduroy, over what counts as a species in for microorganisms, had real implications. And for example, in the early 70s, when the first international environmental movements emerged in response to pollution and other forms of human-driven degradation. For instance, the term genetic resources would later become central to the CBD, the Convention of Biological Diversity, adopted in Rio in 1992, but it actually appears as early as 1972 in the Stockholm report. But when you look at the 1972 report, what becomes very clear is that under the category of genetic resources, the discussions were overwhelmingly focusing on plants and animal genetic resources, along with a new fear spreading at the time, the idea of the human-driven erosion of biodiversity, including through breeding practices for animal and plants. Microbial genetic resources were mentioned, but they were not treated with the same level of concern. That was also related with all these controversies over the true meaning of species related to microorganisms. And by contrast, You can see on the cover of this illustration of the UNESCO's official magazine in 1975, which is in many ways a self-congratulation of the action of the UNESCO in the field of applied microbiology. They could have pictured and celebrated the human victory against pathogens at the time, for instance, the eradication of smallpox. But instead, as you can see, it marks a time of great hope, expectations, and promises. For instance, the use of bacteria for biotechnologies, rather than a fear of the erosion of microbial diversities. And it shows also the difficulties to actually account for the microbial diversity that is still rather difficult to evaluate, and the very small representativity, so to speak, of the ex situ collection, such as the one in Lausanne or elsewhere, this culture collection. So, well, that's a couple of lines that I wanted to highlight as a conclusion. So what I try to do and what I'm trying to do with this work is connecting the history of the medical bacteriological revolution with the trend of this general microbiology, because it's essential if we want to grasp the full dynamics of the discipline from the 19th century. Early international infrastructure mainly contributed by regulating the chaos in names, and the approved list of bacterial names in 1980 was a landmark. And they also, of course, worked on standardizing preservation methods. The first international microbial collection appeared when the very definition of microbial life was very fluid and contested. And what is also interesting that even incorrect or what would prove to be false theories generated knowledge, practices, a connection between institutions and actors that shaped the field's later history. And maybe I go to the last point. The myxomatosis incident also set a key precedent for shadowing modern biosecurity issues. Accidents become a normal condition within the growing routine exchange of microorganisms that was actually developed through infrastructure such as this culture collection in Lausanne. Thank you.
Thank you, Frederic, for this very different perspective on microbiology. There are questions in the room or online, please. Yes.
So thanks a lot. I'm like very right. The tensions that is still reflecting today. What is the current day status of geopolitical tensions around these collections? I mean, now they are aware of good collections in Germany, in Belgium, and the States. I'm not aware of collections existing in Russia, India, and China, but they might advance exist in Japan. There are also. I mean, what is then the geopolitical game, if there is any, around these collections? This is the first question. And the second one, how do they articulate with the Nagoya Protocol that we have then established for sharing resources that are, among others, genetical resources?
Well, thank you for your question. That's a very wide question, and well, that's not exactly what I've been studying, but that's part of the story. So the World Federation of Culture Collection is still alive. It moved from Australia to Japan first, the data center in the 1980s, and then to China I think in 2010. And they were participating to the discussion at the Convention on Biodiversity in 1992 in Rio. And they tried actually to make the case for the question of microorganism biodiversity because as I tried to reflect in one of my last slides, a lot of questioning was very focused on the issue of plant biodiversity and this erosion of biodiversity for plants or animals, which are very different topics. And one of the key issues for the World Federation of Culturation was also trying to develop through new technologies an evaluation of the current state of biodiversity for microorganisms. And you have the first account of this biodiversity of microorganisms showing that actually what is and what was stored in culture collection was non-representative and a very tiny proportion of very large biodiversity in the environment. So the political work that was done by the World Federation of Culture Correction, especially between 1992 and the Nagoya Protocol in 2010, was actually preparing the new rules, especially on the question of the biopiracy and the use of microorganisms from the south by companies or researchers from the north. So that's where they were actually working, trying to have some kind of regulation, shared agreement on this issue of the use of microorganisms that were stored, that were identified and isolated in the south and then used in the north, but allowing at the same time the accessibility for research of this trend. So they did some kind of gray work, behind the scene work to try to control, because there is this question of extraction of microorganisms from the south that will be used by companies without benefit sharing. I was looking for the word for population in the south, so to speak, and so that's where they were actually working.
Any other question? Any question online? No? Yeah.
So, come to think of it, it's kind of hard to imagine that the myxomatosis incident was the only incident. So you went through archives, and I was just wondering if you found any traces of other incidents or escape organisms.
Yeah, good question. Thank you. No, I only found this case. It's actually how I started the project working on culture collection and on this specific center in Lausanne, because I was looking at issues of epizootics and the regulation of epizootics from the Office International des Epizooties, which is the main body, which is still the main body for these questions. Today, I think it's called the World Health Organization for Animals. And no, that's the only occasion, but that I found in the archives. But that's also related to the type of materials that I'm working with as an historian. The archival material that you have in Lausanne have been refound, so to speak, in the late 1990s in a brocante. So the personal paper of Hauduroy then came back to the archives in the Canton de Vaud. And it was the myxomatosis case here is technically, I think, 2 or 3 books, because Hauduroy was really afraid of the bad stigma for the infrastructure and the culture collection in general, but the one in Lausanne especially. And so he had a contract with an agency, I can't remember the name, who was sending him all the newspaper articles. So you have like 3 books of 200 pages of technically all the articles talking about the responsibility of the scientist and him especially with that. So no, through this specific culture collection of Lausanne, I have no other example. But again, that's also my last maybe argument. I think that by fostering the circulation of microorganisms in different states, dry, frozen, or other their materialities, of course, the risk of having some accidental release of microorganism was higher. But no, I don't have any.
Let's hope that's the case. Thank you again, Frederic. Welcome to the online.
Yep.